So now let's shift to the other half. So remember that we can basically think about data using markers to detect selection as coming in two categories. Broad category number one, the last couple of lectures, was using polymorphism data from a sample that detects a single ongoing event or potentially multiple ongoing events at different sites. We're now going to shift gears and talk about divergence-based models, where what I need is data from two or more divergent species, maybe populations if they're far enough apart, but typically more species, or I need multiple species in a phylogeny. So now we're looking at multiple repeated events over a given region, or as we'll talk about a little bit for phylogeny, over a given codon. So now we're shifting gears to those. And the nice thing about those is really only a couple of type of tests, but it also introduces a very broad area about what fraction of the genome is under selection, what fraction is adaptive, and we'll talk about all those concepts as well. Okay? So we can divide divergence-based tests into two things. Population-based divergence test. I basically have a polymorphic sample from a population, and then I've got a DNA sequence from a sister species, and I want to ask if the divergence I see is consistent with the polymorphisms I see. So in the classic case, we look at synonymous sites and non-synonymous sites. That's McDonald-Kreitman. In the HKA test, I look at a number of different genes and ask over those genes, does the polymorphism and the divergence information, do they match up? And so those are population-based divergent tests. I look at different classes of sites within the same gene, McDonald-Kreitman, or compare different genes. That's Hudson-Kreitman, Aguade, the HKA test. That's one type. And what we have there basically is a polymorphic sample and a divergent sample. In phylogeny-based tests, and we get a signal here if within that gene or sequence of interest, I get repeated adaptive substitutions. So a single substitution won't give me much of a single. I need repeated adaptive substitutions, so I have an excess of substitutions relative to background to give a signal. We can also do a kind of a different test, phylogeny-based divergence test. So now instead of having simply two species, I have a number of species, and now what I simply have is I've got a sequence from each species in some phylogeny, and the reason I have some phylogeny is I can determine if events are independent. So I've got, let's say, a mutation appearing several times. It could be clustered in the phylogeny, in which case it's probably a single event, or it could be scattered over the phylogeny, in which case it's probably independent events. And here, I contrast the rate of evolution at different sites within a gene in a phylogenetic context. We talked a little bit about Ka to Ks ratios. We talked a little about codon models. We'll kind of extend upon those here. So these are two different categories. This is kind of medium-term divergence between two species. This is longer-term divergence for a given gene over a number of species. So this requires a polymorphic sample from one species and a DNA sequence from the other to get polymorphism divergence. This requires DNA sequences from your target gene over a number of different species with a known phylogeny. So the first point here to now discuss the foundations of population-based divergence test is that a history of selection alters the polymorphic to divergent, the ratio of polymorphic to divergent sites. And the idea, as we've mentioned before, we can measure divergence and measure polymorphism, and under the standard neutral model, we can have expressions for it. So for example, the level of diversity measured by, for example, nucleotide diversity is 4Ne mu I, where mu I is the mutation rate for the class of events you're looking at. The divergent rate is 2T mu I. So if I've got a species that's diverged with a common ancestor T generations ago, that's the expected divergence. So for a given gene, if I look at the ratio of polymorphism to divergence, if you'll notice here, the mutation rate, which is unique to that gene or that sequence, cancels, and I get a number, and so that ratio should be very, very similar as I go across different categories or different classes of genes in the same comparisons, because basically the mutation rate, which is what differs between the classes or genes, that cancels, and I get something here that should be relatively constant. If instead of looking at diversity by measuring polymorphism by measuring pi, if I measure segregating sites, I get something very similar. The only difference now is I get this AN, which is a sample size feature. But again, if I have a number of genes from the same sample, that ratio should be the same. So under the neutral theory, there's this prediction that the polymorphism to divergence ratio should be constant over genes or over different regions within a gene, such as synonymous versus non-synonymous. And we mentioned the classic example of this, which is Kreitman and McDonald's test for alcohol dehydrogenase. So what they did was the comparisons here where I have a gene of interest, alcohol dehydrogenase, I break that up into two parts. Part number one is the so-called silent or synonymous sites. Part number two is the replacement or non-synonymous sites. At the second site, a change in a nucleotide will change the amino acid. At the first site, a change in the nucleotide will code for the same amino acid. The idea is the synonymous or silent sites are largely neutral, at least to a first approximation, so they have a certain effective mutation rate. If I go to the polymorphic site, the non-synonymous sites, they have the same underlying mutation rate for nucleotides, but they have a different acceptance rate because a lot of those mutations are deleterious. So if I look at the ratio of replacement, that is fixed differences versus polymorphism in silent sites and fixed versus polymorphic, these ratios should be the same if basically both these are being influenced by neutrality. Now the key here is for the replacement genes, I imagine deleterious mutations do occur, but they're very quickly removed, and the net result in that is these sites basically have a lower effectively neutral mutation rate than the silent sites do, and that results in them having a lower value of here. But if I look at the ratio of polymorphism to divergence or segregating site to divergent, that difference is that mutation rate, which differs, the effective mutation rate between synonymous and non-synonymous, that cancels, and I should get a similar quantity. So those ratios should be, that ratio and that ratio should be quite similar. Fisher's exact test shows it's highly significant. And furthermore, we can basically go in and ask, given this ratio and this number of polymorphic sites, what's the expected number of substitutions I would see? And that number is 1, so I basically have, actually a little bit less than 1.8. So I actually see an excessive number of replacement substitutions, in this case 6. If you believe that number, it then says of these 7 substitutions I see, 6 out of 7 are adaptive, more than I expect. So in this particular gene, 86% of these replacements are adaptive. So we're going to take that idea and flush it out to talk more generally about fraction of adaptive sites throughout the genome. And that's the basic idea behind McDonald-Kreitman. The ratio for silent sites of divergence to polymorphism should equal the ratio at replacement sites of divergence to polymorphism if the standard neutral model holds. Okay? That's the basic idea. So that's one category, divergence-based tests. We're going to come back and talk about those in a second. The other category is a history of selection should argue the ratio of silent to replacement substitutions. So here I'm contrasting polymorphism versus divergence. So I've got two species. Now I'm going to move to a phylogeny and basically contrast over the phylogeny. In that phylogeny, how rapidly do third base positions change? How rapidly do replacement positions change? And the basic idea is that negative selection is quite common because the pattern we almost always see, if you go over any gene, over that entire gene sequence, it's almost always the case the rate of substitutions is lower at replacement, that is non-synonymous sites, than it is at silent sites. And the argument is that a large number of the replacement at silent sites are deleterious. They get removed very quickly. The only thing then that gets substituted are, under the neutral theory, are then effectively neutral sites. And therefore, there's a lower mutation rate, effective mutation rate, at replacement sites because they both sites have the same amount of mutation coming in, but the silent sites accepts a large fraction of those as being neutral, the replacement site accepts a much smaller fraction. Therefore, the rate of evolution at replacement sites is much slower than the rate of silent sites. And we use Ks to denote the per site silent substitution rate and Ka to denote the per site replacement substitution rate. And the ratio Ka to Ks is often called omega. We expect that value to be less than 1. A value bigger than 1 is strong evidence of selection. Although, as we'll see, there are other reasons you can get that value as well. So I already mentioned the classic study by Austin Hughes, who unfortunately passed away a couple of years ago, and Masatoshi Nei, and this was looking at MHC1 and MHC2, major histocompatibility. These molecules that basically have certain configurational shapes, and those shapes grab antigens. If you look over the entire sequence, comparing humans and mice, what you see is you see a much higher rate of substitution at third base positions, that is silent, than you do at first or second, that is replacement, over the entire gene. But what Austin and Hughes did was they took the protein folding data, on that protein folding data, categorized what they thought were critical sites for antigen binding, then put those sites aside, looked at those codons separately from the rest of the gene, and looking at the codons for those antigen binding sites, showed that they actually did have a Ka to Ks ratio bigger than 1. That is, at these particular antigen binding sites, you had replacements at protein coding that result in amino acid changes were quicker than those that resulted in silent changes. And that's strong evidence for directional selection in those sites. The way they could do that is they could break that protein sequence into important residues that we think are under selection and not important, and then test those categories separately. It's not based on any spatial information, because as I mentioned, when a protein folds, it's like a big rope, and so a site over here and a site over here may actually be in contact to make that antigen binding site, but you wouldn't spot that on the one-dimensional protein sequence. So that's kind of the classic example of showing elevated Ka to Ks ratio, but it required you to know a lot, which is namely the protein folding, and from the biology know what sites on that protein folding you think are interesting. So what people have done is they've then, as we'll talk about, replaced this then for codon models. Yeah. Sure. That's correct. Basically, higher fraction of deleterious mutations at replacement sites than at silent sites. Well, no. So it's larger one is a more conservative estimate sign of selection. So if a Ka to Ks ratio is usually 0.4 and you see one of 0.7, you could take that as evidence of selection as well. But the broader way to think about that is you're seeing an excess of when it's bigger than 1. It's clearly an unambiguous signal. Well, it basically means that the rates are faster than at the silent sites, which is sort of bulletproof, right? Because you obviously expect, everything else being equal, replacement sites to have a lower turnover than silent sites. These are a bit faster. So one unstated thing is these divergence-based tests are biased towards conservative sites. And this is something I don't think is well appreciated. What do we have to do to run these tests? We have to align sequences. Aligning coding regions is pretty straightforward. We can align coding regions. However, if I have non-coding regions, aligning them is very problematic. You need common features. Common features mean conservation to line them up. So these methods are usually only applied to coding regions. You can apply them non-coding regions, but it's a biased sample because you're only applying it to non-coding regions, which are under strong sequence conservation, so you can line them up. So there's this bias in using these sort of approaches that requires you to align sequences. Protein coding sequences do that easily. Regulatory sequences, which may be extremely important, aren't necessarily conserved, so it's very hard to line them up. So we're looking at a biased set when we move outside of protein coding regions. Just be aware of that. Yeah, question. Sure, that's a great question. So in a non-coding region, there's no such thing as synonymous and non-synonymous. You basically break it into two categories. So for example, what you can basically say is you can say every even codon versus every odd codon. Just looking at some way to get some comparison. If you go to the HKA test, you don't have to worry about that. HKA test just looks at in that entire gene polymorphism to divergence. But you can take McDonald-Kreitman and extend the basic logic from two categories to as many different categories as you want. So you can describe the sequences two ways, like synonymous, non-synonymous, or maybe transitions, transversions. There's other ways you can look at that same data. So you're right, with non-coding regions, there's the issue of how do you classify them. But in an HKA test, you just care about the gene, not about synonymous, non-synonymous. So you can certainly put them in an HKA framework. So a really fundamentally interesting question is what fraction of the genome is under selection? So humans are 3 gigs. How much is our coding sequence? 33 megabases. You take all our protein coding sequences, shrink the introns down, line them up, it's about 1% of our entire genome. Obviously, we have a lot of stuff in there like ALU elements and other dead mobile elements in there. And so a really interesting broad question becomes, what fraction of the genome is under selection? Now, one way you can approach that, and it gives you a lower bound, is to ask what fraction of sequences are conserved. So look at conserved sequences, you need an outgroup. So for example, when you look at, mice, humans, and dogs, what you basically see is the net result is that about 6% of our genome is conserved over those timescales. That's 6 times more DNA than is in our coding regions. Now, part of that is, boundary regions. So when I say coding region, I basically mean if you look at processed messenger RNAs. So obviously those introns would be regarded as being non-coding, even though they're transcribed. There obviously are signals in some of those, but the bottom line is one initial estimate based upon long-term conservation, so mice, dogs, and humans, is about 6% of our genome is conserved. 6 times more than the actual nucleotides used for coding. Now that's obviously a lower bound because important sequences which are conserved can still turn over, and so those conserved sequences might be found in the great apes, but you wouldn't find them, for example, in dogs. So additional studies basically went in and looked in a little bit more detail, with those in a second. The same logic in Drosophila, and between 40 and 70% of the melanogaster sequence seems to be constrained when looking at sister species. That's about twice as many as the coding region. So it's got a smaller genome, codes roughly the same amount of genes, so a higher fraction of the genome is involved in coding. But again, both these studies show that at least half of the conserved DNA in Drosophila in humans, and in many cases much, much more, is non-coding DNA. So you can go a little bit further, very interesting paper here. What they basically did was they looked at species which were further and further apart and looked at the decay in conserved regions and then used those to extrapolate and ask how much conservation am I missing by looking at really deep groups. What they basically found is the frequency of conserved DNA in humans is between 200 and 300 megabases, so on the order of about 7 to about 10%. They finally refined this to about 8%. So 7/8 of the conserved DNA in humans is non-coding. In Drosophila, again, you get a value of a little bit more than 50%. So a lot of the DNA in us that's not coding is conserved. In humans, several times more non-coding DNA is conserved than coding DNA. It raises all sorts of really interesting questions. How strong is selection in these regions? How often do adaptive mutations arise in non-coding regions? What fraction of deleterious mutations are attributed to these regions. Think about background selection. We think about background selection as deleterious mutations rising in coding regions. If non-coding regions are important, there are a larger target size, a higher fraction of deleterious mutations may be arising in those regions. So this sort of silent signal of conserved regions outside of coding is kind of like the bottom of the ocean. It's an area that's only now starting to be explored. So people often focus on coding regions. Non-coding regions are maybe more important. Most GWAS hits go to non-coding regions. And I mentioned a simple example. Suppose that the computer over there is a gene whose expression level changes and influences, let's say, height. Suppose, though, that that gene is controlled in some level by small RNAs that bind to control regions. I now have a mutation over here on a different chromosome that takes a random sequence. A one nucleotide change now means I've got a little bit of similarity to the small RNAs that bind over there. So some fraction of the small RNAs that bind over there now bind over here loosely. The amount of small RNAs binding over here goes down a little bit. The expression level changes slightly. If that's dramatic, the change in expression for this gene that influences trait is determined by a non-coding region over there. And that would be an example of a non-coding region that's not even conserved, just random sequences that happen to capture or titrate enough of these small RNAs off to give you differences in regulation. So a big unresolved issue is what type of selection is going on and how important is selection outside of coding regions. I think the correct message is we have yet to fully appreciate how important it is. Just like we're only now starting to appreciate just how important small RNAs are, because we didn't see them much early on, we thought they weren't important, we're now starting to appreciate that a much larger chunk of human DNA is probably under selection in simply coding regions, and obviously lots of interesting things can happen there. It's harder to find, and part of the reason it's harder to find is if I have a protein coding sequence, I've got a base pair change, I can tell you, oh, that changes the amino acid from this to this, that's a radical substitution. If I've got a change in a conserved region, and it's outside of these really obvious, consensus sequence regulatory sites, I can't say much about what that single nucleotide change is expected to do. But it could be quite important. So one thing to be aware of is kind of one of the unknown frontiers is just how important selection in these non-coding regions is. It's clearly important, no question about that. Just how important it is in terms of deleterious mutation load, adaptation, is a very unresolved issue. Yeah, question? That's a great question. So the question basically is, what do I mean when I talk about conserved sequences? This first row is a conserved sequence. A mutation there which is deleterious, a mutation could arise which makes it deleterious, that affects background selection. So that would have an impact on the genome through the effects of background selection. But it also could be this region is conserved because it's involved in regulation, and a nucleotide change in there now may be adapted to a certain trait. So the key thing is if I have a region that is influenced by selection, it could also then be a region where mutations arise which are under positive selection. When we think about conserved sites, we mainly think about having a functional role, therefore don't change it. That affects background selection. That affects our overall deleterious mutational load. But it also means that in rare cases in there, just like a protein coding sequence is conserved, you can have rare mutations which are beneficial. In the conserved regions, you could also have rare mutations which are beneficial as well. We simply don't know. The bottom line is, if you imagine the genome as being this giant pie that's 3,000 megabases large, we have this little sliver that's about 30 megabases, this protein coding genes. We've got this bigger sliver, about 300 megabases, that are other sequences simply under selection. So those are sequences which have some functional role because by definition, if they're conserved, means you change something about them, it impacts something in the cell. It may be where chromosomes bind, it may be chromosome folding, it may be long-range methylation, it may be gene expression. We really don't know. All we know is that for whatever reason, this chunk is conserved. That contributes to our deleterious mutation load, but it also means since those sites are involved in some functional purpose, some fraction of them could also rarely generate advantageous mutations as well. But not at all. We're simply, well, we know that because they're conserved, that most mutations are deleterious. But that doesn't mean all mutations are deleterious. And it simply means from the big picture that we have to look beyond protein coding genes, ask broad questions about the role of adaptation. Because if these things have a functional role, some changes in those functional role may be beneficial. Probably under specific situations. Just like if I've got a protein coding sequences, almost all changes are bad, but a few changes are quite good. That's how we adapt those proteins. The same thing for conserved regions. Most of those are probably, but the point is, it's also a source for generating deleterious mutations, which affects background selection, which alters the pattern variation across the genome. So the bottom line is, we can't simply think of the genome as coding regions floating in a sea of stuff we otherwise really don't care about. There's a lot more stuff out there. In fact, in humans, probably 8 times as much, 7 or 8 times as much, targets where mutations can have an impact on fitness in non-coding regions than in coding regions. In Drosophila, it's about 50%, but part of that is because Drosophila has a much smaller genome and has roughly the same amount of coding DNA. But even in Drosophila with a much more compact genome, at least half of the remaining sequence seems to be under some type of selection. So just a background thing to think about. That's an expanding area in evolutionary biology and genomics. So now, any other questions or comments? Let's now talk about the 2 main categories of tests for divergence based upon a polymorphism sample and a divergence sample. Test number 1 is HKA, Dick Hudson, Marty Kreitman, Aguade, the HKA test. So you see Marty's in 2 of these. The University of Chicago is where this was put together. So they proposed the first test to jointly use polymorphism and divergence data. McDonald-Kreitman takes a given gene and looks at synonymous, non-synonymous. The HKA test basically takes a series of genes and looks across gene 1, the ratio of divergence to polymorphism, across gene 2, across gene 3, etc. So the HKA test can be applied to any type of sequence data, not just silent versus replacement, and that gets at your concern about if I've got non-coding regions, how can I classify them as silent versus replacement? I can, for example, have 5 protein coding regions and 1 non-coding region, and do HKA tests by looking at polymorphism to divergent ratio at the coding genes and also the non-coding genes. Again, the issue for non-coding genes is they have to have enough conservation so I can line up homologous regions and actually ask how many divergences did I see. And that gives you a bias set. Yeah. Good question. So you can basically sample a couple of different ways. When these were first done, they had a small sample from Drosophila, because sequencing was hard, and took 1 sample from another species of, so a small population sample from melanogaster, and then 1 sequence in that gene from another species. You could sample both populations. As we'll see in a little bit, for McDonald-Kreitman, and as we've seen before, which population you use as your polymorphism sample can actually change your results. So I think that the way to go if resources aren't an issue is to have polymorphism samples from both populations and combine those samples together and use that combined sample as your polymorphic reference point. I think that's So the idea is that population structure would jointly affect, presumably, under neutrality, all those polymorphism samples the same way. And so that kind of cancels out when you look at the ratio of across those, right? We'll see an example in a second where you've got to be careful with mitochondria and chloroplasts. We'll come back to that. And in that case, basically, they have a different population structure than autosomes because they're transmission. We'll talk about that. So basic idea behind HKA, it's a very simple model. You've got two populations. So population A has an effective population size. Population B has some unknown parameter delta. So it's either larger or smaller. So parameter number one is this fractional difference. If delta is 1, they're the same. Parameter number 2 is they've been separated by some amount of time from an assumed common population. So that's two parameters, constant over all genes. Difference in effective population size, time of divergence. Then I have L unlinked loci, and at each unlinked loci, what I have is the expected number of segregating sites is simply given by the mutation rate in species A, effective population size. In species B, I now have a slightly different, so I can write that then as delta times this expression here. So if I have 5 genes, I've got 7 parameters. I've got this common effective population size, I've got this common divergent time, and for each of the 5 genes, I've got a different theta I. So you put all that together, and you get expected number of segregating sites for gene I in population A, gene I in population B. These things just depend upon the sample size. The expected divergence for gene I, that basically just gives you that expression, variances for that. And you can put all those together and then get a test statistic. You can estimate, so what you basically have is you've got 3 L observations. What are the observations? For each of your L genes, divergence in polymorphism in population 1, polymorphism in population 2, divergence between the two. So you've got 3 L observations, 2 polymorphism, 1 divergent for each of your L genes. Then you've got things to estimate. You've got the common effective population size difference delta, the common divergence time tau, and then you've got, for each gene, you've got a different theta value. So because you've got this set of equations, you can then go ahead and then estimate the common tau, the delta, and then the theta i's for each of the genes, and that basically then gives you 2L minus 2 degrees of freedom. Number of observations needed to do estimation. So you then construct a statistic here, which is very much like a chi-square, and this is the effect of a given gene. So for a given gene, you compute its level of polymorphism based upon segregating sites from its expected level in population A, same in population B, divergence, scale each by their variance, and then sum this over all genes and get a single summary statistic. The nice feature about this, if your summary statistic is too large, that is, it's significant, you can then go back and ask which gene was driving that. So you get an overall summary when you sum over all the genes, but then you go back and ask which gene is giving a larger signal. Just like if you do a chi-square test for goodness of fit on a contingency table, you can go back and compute chi-square values for each particular cell and ask if any particular cell in that contingency table is basically driving most of the significance. Same thing here. So here is an example of when they first applied it, and this shows the quality of data they had back then. So they had 2 loci. They were comparing silent sites and 4 kb of the 5' flanking region in ADH. So they had sequencing data from Kreitman for the ADH gene itself, and what they basically did was they took 81 alleles from melanogaster and a single allele from Seychelles. When you do that, basically, this term goes away and you make an adjustment for it. It's not a big deal. So based upon sequencing data, they had 210 differences in the flanking region and 18 differences in the silent sites. They then used restriction data to look in melanogaster and found that 9 of the 414 5' flanking sites were variable, and 8 of the roughly 80 silent sites were variable. You can then plug these numbers in. The book goes through those calculations. You then get a test statistic of about 6. The probability that chi-square with 1 degree of freedom, sorry about the funky fonts here, bigger than 6 is a little bit more than 1%. So these data show a significant departure from the equilibrium neutral model. Again, equilibrium neutral, you have the same issue with site frequency spectrum tests. The departure could simply be because the model is not at equilibrium. Yeah, question. Because part of the reason is we don't have polymorphism data for Seychellii. So we've adjusted that way. Yeah, good point. Good catch. Glad you're paying attention. So knitting and paying attention at the same time, right? Yeah, no, that's fine. No, a lot of people do that to a lot of people. My wife does that. She basically does braiding, and it focuses when she's thinking about stuff. But for some reason, when she braids, she can never hear me talk to her. She can focus on everything else. But like, say something. Except if I mention shoes or purses, those seem to be magic words that catch her attention. Let me show you then where you can have issues with this test. So this is a test. It's assuming unlinked loci. So if I look at a mitochondria or a chloroplast, that is a single locus, because all those sites are fully linked. So a nice study here. Here's a lovely plant they were looking at. And what they did was they looked at 4 non-coding regions of the chloroplast. Those aren't 4 sites. That's a single locus, because they don't recombine. Same for mitochondria. And likewise, same for multiple genes on chromosomes with very little to no recombination, let's say the Y or a dot chromosome. Then they looked at 2 unlinked autosomes between these 2 species of this genus, and there's kind of what it looks like. And they got a highly significant value. And by looking at this statistic here, they saw that most of the signal here came from the large value in the chloroplast signal. So they thought about this a bit. Then they went ahead and they did FST. You can do FST on the chloroplast. It's a haploid. You can still do FST. And on the autosomal genes, what they found was FST in the chloroplast was about 0.55, and it was 1/10 of that in the nuclear gene. Now, this is not at all uncommon, because remember, transmission, mitochondria and chloroplast are maternally transmitted. Anybody know of an exception? There's one I know where the male transmits both the mitochondria and the chloroplast. Redwoods. No idea why. Don't ask me why. But what you have is sex-specific migration. And this can structure the population, because pollen can flow very different from seed. Males often disperse more widely than females, and so you can get very different population structures. Remember years ago, an interesting talk, if you look at the Channel Islands off the coast of California and look at mammals there, they are essentially the same autosomally as on the mainland, but they're quite different mitochondrially. Again, differential gene flow can occur because you have differential gene flow in the sexes. Another really interesting area that comes in as people start looking at genomic differences for species is, people remember Haldane's rule? Who's heard of Haldane's rule before? See how your training is? Haldane's rule is that you often can have sibling species where if you cross them, you see one sex is viable and the other sex isn't, or what you see is one sex, both sexes are viable, but only one sex is fertile, the other sex is not fertile. Haldane rule says it's almost always the case that the viable or fertile sex is the homogametic sex. So in mammals, that would be females. So in mammals, if I've got hybrids, since females are viable, mitochondria can flow across species barriers. Now let's think about birds and Lepidoptera. In birds and Lepidoptera, the heterogametic sex is males or females. Heterogametic sex. The homogametic sex, what about bumblebees? It's mammalian? Gotcha. Okay. Yeah, so most time not perhaps. Okay. So, but anyhow, there are groups of organisms where it's flipped around. So, for example, in butterflies and moths and birds, mitochondrial gene flow is blocked because those females are going to be inviable or infertile. So, genes on mitochondria and chloroplast, they can have different population structure within a population, and they can have different rates of flowing from autosomal genes between closely related species. So here, you can do corrections to basically correct for this. You can do the corrections for segregating sites and for divergence. When you make those corrections, the HKA test is non-significant. So you get a high HKA test here because basically you've got a different population structure for the organelle genes than the autosomal genes. Just a cautionary point to be aware of. Likewise, you've got to treat the mitochondria and chloroplast as a single locus. If you've got information, mitochondria is one locus, chloroplast is the second locus. So together they're two loci, but no matter how many genes you have in the chloroplast, that's just one gene in the HKA test. So that's HKA. It was the first test that was put out there. It's got the nice feature. You could apply it to any sort of sequences you want. And it's a comparison test. And you can also use it to ask which particular sequence is giving you the signal. Any questions about that? The most popular test is the McDonald-Kreitman test. Same K as in HKA. One of the most widely used requires a polymorphism sample from one species, or you could do both, and a divergence sample between species. It's robust to demography, provided the effectively neutral mutation rate is the same in both polymorphism and divergence phases. We talked about that earlier. We'll come back and talk about that in good detail as well. So it's kind of like an HKA test, but instead of contrasting across genes, it contrasts silent and replacement within a gene. And that turns out to give you a lot more advantages. So here's the basic idea. The basic idea is if I have a effectively neutral mutation rate, the polymorphism measured by nucleotide diversity is 4 NE mu. If I measure a number of segregating sites, it's this times a constant. It changes with the sample size, no big deal. And the divergence is given by 2T mu. That's the effectively neutral mutation rate, which basically means if S is my selection coefficient, if this is satisfied, those alleles behave as if they're neutral. So McDonald-Kreitman assumes that there are genes under selection, but almost all the mutations are deleterious, and they're so deleterious that when they arise, they don't contribute to divergence, and they also don't contribute much to polymorphism. So what you're left with then is when a mutation arises, the silent sites basically have almost all those mutations being effectively neutral, but a much smaller fraction, F, of new mutations in the coding regions are, the replacement sites I should say, are neutral. So mu is the mutation rate, the neutral mutation rate at silent sites. F times mu is the effectively neutral mutation rate at replacement sites. So you can then look at what the ratio of divergence at replacement over silent sites is, and the numbers here simply account for if I have a codon, 2/3 of those sites on average can change amino acid, 1/3 can change that. Look at the polymorphism ratio, the same thing. The net result is these two ratios are the same. So under the equilibrium neutral model, the ratio of divergence in replacement over silent should equal the polymorphism in replacement over silent. You can look at that another way and basically express it and get the same result. You can basically modify this to get several different things. So we have this expectation of ratios being equal. Under the neutral model, because even though the mutation rates differ, if I look at the right expressions, things cancel. So over here, both these ratios have F in them, so the common F cancels. Over here, I've made the ratio of polymorphism and divergence, so F cancels within here. So both these scenarios give you the same thing of ratios being equal. So either the divergence over silent equals a polymorphism of replacement over silent, or the ratio of polymorphism to divergence under replacement equals ratio of polymorphism divergence under silent. And that's the basis for the McDonald-Kreitman test. If you look at this 4NE term that comes into polymorphism, you can actually replace that by a term that looks at the total amount of time on that coalescent branch. That allows you to have non-equilibrium population. The ratio of those two cancel, so that's removed. So it's often stated that the HKA, the McDonald-Kreitman test, is unaffected by population demography. That's because you can have non-equilibrium populations generating your polymorphism. It's the fact you're looking at the ratio of those two polymorphisms. That common effect that influences both things in the same gene cancels and goes away. If the effectively neutral mutation rate is the same in the polymorphism phase and the divergence phase, it's completely unaffected by demography. As we'll talk about a little bit, demography can actually influence that, and that can change the test. So you very often see data presented in this form. This is called the DPRS table. Divergence, polymorphism, replacement, silent, in a clockwise fashion. So when people talk about using MK data, they mean you generate a table like this, and then you analyze that data. Okay? So divergence, polymorphism, replacement, silent. And here's an example of the Phrygian gene in Arabidopsis involved in flowering. And if you look at across the entire gene, you see just a little bit below, not quite significant. So you basically look at the ratio of replacement to polymorphism at silent, ratio of replacement, by fixed I mean substituted. So substituted to polymorphism, substituted to polymorphism at the replacement, and that's just barely below significant. If you look at exon 1, it's highly significant. You combine exon 2 and 3, it's not significant. So what you can basically show here is almost all the signal is coming from exon 1. How could we interpret these results? Well, one way you can interpret the results is if I have a relaxation of selective constraints, that could give me a positive signal. However, you have a nice internal control in that if replacement polymorphisms, selection constraints have been relaxed so they're not nearly as deleterious, you'd expect that same pattern to be seen in exons 2 and 3, and you don't see that. So the net result, what it looks basically like here, is there's an advantage to having replacement polymorphism in exon 1, and the notion is that the majority of these are loss of function mutations, which result in early flowering. You have lots of function in exons 2 and 3, apparently it doesn't result in early flowering, just loss of function in exon 1. So you have an excess of these because there's apparently selection for earlier flowering, and the interesting thing here, this goes back to soft sweeps, is there are many different ways you can generate that early flowering in the same gene. You simply have to knock that gene out in exon 1, and there are lots of sites to do that. So that's a nice example of using the McDonald-Kreitman test. An obvious question becomes, if I get a significant result, what is driving that? Relative to my neutral standard, do I have an excess number of replacement polymorphism, or do I have an excess number of replacement fixations? The way you categorize that is the neutrality index. Here's the ratio under silent sites. If you have an excess number of polymorphisms relative divergence at the non-synonymous sites, the neutrality index is bigger than 1. too many polymorphisms. If you have an excess number of divergences, fixations relative to polymorphisms, the neutrality index is less than 1. So neutrality index bigger than 1 is taken as two things. Well, number one, too many polymorphisms. And the standard interpretation for too many polymorphisms is you've got either those polymorphisms are favored or they're deleterious. But they're weakly deleterious. So they're weakly deleterious. They segregate. They're not so deleterious they get knocked down, but they're too deleterious to become fixed. So neutrality index bigger than 1 is usually taken as an indication of deleterious replacement polymorphism segregating. Neutrality index less than 1, too many replacements relative to polymorphisms, is usually taken as evidence of adaptive fixations. So you go back here to the Phrygia data, and the neutrality index for exon 1 shows a massive increase in the amount of polymorphism relative to divergence. We've put an adaptive spin on that. Doesn't have to be, but I think in our case you can make a point because these polymorphisms result in loss of function, which gives you early flowering, which in this case is favored. But in general, you take your comparison, a neutrality index bigger than 1 usually means you've got deleterious segregating alleles, because McDonald-Kreitman basically assumes that if you're deleterious at all, you get knocked out. You don't see it. If you're weakly deleterious, you contribute to low-frequency polymorphism, but not to divergence between species. So examples of this, so here's Peter and Carlos are two of the young investigators doing a bunch of work. So Princeton, and I think Carlos is now at Stanford. So here's one of Peter's early studies. He looked at 35 coding and 153 non-coding fraction from a population of Drosophila X chromosomes as my polymorphic sample and compared to a Drosophila simulans as the outgroup. Then what he did was, instead of looking at a single gene, he looked over a bunch of genes and amalgamated them. So our standard is going to be the silent sites here. So in this sample, what he saw was 502 across the entire silent site, 502 polymorphisms in this sample over all these sites in melanogaster, and he saw 604 fixations between this large amount of sequence from melanogaster to simulans. If you go to coding regions, lump them together, you saw 115 polymorphisms and 260 fixations. So that's the standard McDonald-Kreitman, silent and replacement. The difference is instead of a single gene, I'm lumping over genes. And as we'll see, this is okay, but there can be some problems. But he then went further, then asked about non-coding, 5', 3'. So here, for non-coding, he saw roughly 2,400 polymorphisms and saw roughly 3,200 fixations. You can compare this with this, that with that, and ask if there's an excess. Same for 5' untranslated regions and 3' untranslated regions. Now you might ask, what's this category here? Minus singletons. The concern with a lot of the McDonald-Kreitman test is some of your polymorphism are rare deleterious alleles which are kind of floating around and going to get knocked out of the population. They'll never contribute to fixation. But because they're around there, they basically give you an improper read of what your effectively neutral mutation is. So you remove them because they're deleterious, and when you do that, you'll notice that the Fisher's test compared to silent is significant if you look at all sites, but much more significant if you throw the singletons out. So this basically shows you how you can apply McDonald-Kreitman-like thinking to non-coding regions as well. Carlos did a similar thing where he basically sequenced a little bit under 12,000 genes in humans and contrast that with chimps. SNPs are basically polymorphisms. So he has this HK table. You can then compute a neutrality. So if you do this, the overall p-value is quite significant. you ask, okay, where's the discrepancy coming from? The neutrality index basically shows you have an excess of replacement polymorphism to replacement fixations. And again, the interpretation of neutrality index bigger than 1 is usually that there are deleterious, you have an excess here because you've got deleterious mutations which are segregating, which aren't strongly deleterious, so they segregate, but they're too strongly deleterious to become fixed. And you typically see in humans neutrality indices bigger than 1 because there's lots of rare, apparently deleterious mutations segregating around. So another application of HKA, both Peter and Carlos here, their HKA applications aren't for a single gene, but amalgamating data over a bunch of genes. That's good and bad. You can amalgamate a small signal, so a little tiny signal that wouldn't be obvious in a single gene applies over a bunch of genes. As we'll see, though, there's some potential issues with doing this. If I look at HKA, McDonald-Kreitman for a single gene, the replacement and the silent sites are right next to each other. They share the same coalescence, and that corrects for itself. If I look at these things of lumping over a whole bunch of genes, you could be comparing different coalescences and having a bit different result. It's more of a problem when you look at here. So, yeah, again, one of the problems in applying the HKA, the McDonald-Kreitman test, is that how do you account for an inflation of polymorphism from deleterious alleles, slightly deleterious, which aren't going to contribute to fixation? Because McDonald-Kreitman basically says the alleles that contribute to polymorphism also have the same chance of contributing to fixations. If you have slightly deleterious alleles, you can have a fraction that contribute to polymorphism, but will never get fixed. And so what we'll talk about now are ways to adjust that. But before I do, questions. So we talked about HKA test, and now we talked about the basic idea of McDonald-Kreitman, and now showing how it expands to kind of genome-wide issues. So as I mentioned, one issue with McDonald-Kreitman, besides segregating rare deleterious alleles, the second issue is this. Your effective mutation rate is F times mu for your replacement sites. In McDonald-Kreitman, you're assuming that that value of F is the same for polymorphisms as it is for divergences. We've already seen one way that can break down, because you could have alleles which are effectively neutral to the point that you'll see them as being polymorphic in a sample, but those same alleles aren't effectively neutral in the chance you'll never see them substitute. A deeper issue is, suppose I've got a population size here that's large. That's my polymorphic sample. The divergence, though, happens when the effective population size is small. The effectively neutral mutation rate is larger in the small population. Why? Because F here basically is the fraction of all sites where for any absolute value of S is less than 1. As I change my effective population size, that fraction can get larger or smaller. And here's one way to see that. I can actually look at the estimated constraint. So F is the fraction that gets through, 1 minus F is the constraint, and this is what Peter did a while ago. He basically plotted the observed constraints as a function of effective population size. What you see is the constraint gets bigger. That means that's 1 minus F gets bigger. That means F gets smaller. So as your effective population size gets larger, your fraction that you let through gets smaller, and that can compromise your McDonald-Kreitman test. And I gave you an example here. We saw this one earlier, looking at pigmentation genes. If you take Africa as your sample, there's very strong selection here against any replacement sites. You go to Europe or East Asia, there's very weak selection. And so basically, even though the divergence is the same, that's almost significant. That's not at all significant. And it depends upon your polymorphic sample. Here, you have different type of selection, so any replacement polymorphisms were bad. Here, there's relaxed constraints on your sample, so replacement polymorphisms are relatively neutral. I mentioned this about recombination. This is a point that Peter basically made. If you look at a standard McDonald-Kreitman, here's your gene. I've got groups A and B. I've got replacement and silent. And in that gene, they're concatenated together, so the coalescent structure shared by A is pretty much the coalescent structure shared by B. So comparisons within a gene, for all intents and purposes, both the replacement and the silent sites share the same coalescent structure. If I'm, for example, contrasting 5' UTR regions with silent sites in a gene, now my comparison, here are the silent sites in the gene, here's the 5'. Those could have different coalescent structures because they're not fully interdigitated, and that can potentially compromise the test as well. So just something to be aware of. So we're going to come back a lot more after the break and talk about using McDonald-Kreitman type tests to estimate the fraction of adaptive substitutions. But for now, that's what I wanted to give you the overview. And the key idea is what the basic structure of the HKA and the MK test are. HKA, I look at polymorphism and divergence data over a series of genes. McDonald-Kreitman, I look at polymorphism versus divergent data, usually over sites within a gene, or I'm comparing two categories. Polymorphism divergent at silent sites, polymorphism divergent at 5' or non-coding sites. We have this concept of a neutrality index. It's basically the ratio of polymorphism to divergence for replacement sites divided by the neutral standard. If that's bigger than 1, it indicates an excess levels of replacement polymorphism. If it's less than 1, indicates an excess level of replacement fixations. One of the constraints you can have in the McDonald-Kreitman test is it's independent of demography for looking at the polymorphism, but it depends upon demography if your effectively neutral mutation rate is different in your polymorphism phase versus being in your divergence phase. Those are the key concepts about the population-based divergent tests. So questions or comments on those? The other category of tests is phylogeny-based divergence tests. And here we're looking at, in a phylogeny, looking at a large number of species comparisons and simply asking if replacement sites evolve at the same rate as silent sites. And we basically have seen these before, and this ratio omega of Ka to Ks, per site rate of evolution at replacement sites, per site rate of evolution at silent sites. There are a couple things that can compromise this test. So like we basically said, a value of omega bigger than 1 is usually taken as evidence for selection. You're seeing faster rate of replacement substitutions than you are silent substitutions. We'd expect the opposite because replacement substitutions presumably have more constraints, therefore a smaller fraction of mutations are neutral. Number 1 is kind of interesting. Mutation conversion bias. So genes have a bias to have conversion from AT to GC. So people know what gene conversion is? So when you have recombination happening, we think of recombination as an exchange of outside markers in some region. But in reality, when you make recombination, you get this unusual structure called the holiday structure, where you get 2 DNA molecules intercatenated. When you resolve that intercatenated structure, you either get exchange of outside markers or not, but you also get what's called heteroduplex DNA. You get a DNA duplex where the 2 strands are almost complementary, but contain different information. It's a mismatch. That mismatch can be repaired by either replication. So if I've got a blue and a red strand, the blue strand replicates to 2 blues, the red to 2 red because of semi-conserved replication, use the backbone, or repair. So I could have a blue-red strand, and it gets repaired in a region back to red. So I have the 4 products of meiosis. I would expect to get 2 red and 2 blue, but you can often see 3 red and 1 blue, or 1 red and 3 blues. That's gene conversion. It actually happens a lot more than people think, and there's an intrinsic bias to it, so you can get kind of a sequence tendency so that replacement have a lower GC content, and there's more opportunities for AC to be changed to GC from conversion bias and get that. So the key part about this, and you can look at references in the book, is you can get an inflated Ka to Ks ratio not from anything to do with selection, but simply from aspects of DNA repair, which simply happens because conversion bias tends to be in a specific direction, and that also tends to match the base composition more favored by replacement sites. The second interesting feature is if I have a weak selective constraint that opposes a mutational bias, that can actually accelerate the rate of evolution over a neutral site. And the reason is the mutations take you in the wrong direction, and selection drives you back to these continual cycles of fixation that actually gives you a higher rate of fixation than neutrality. So both these things are potential things that can inflate the ratio. They're relatively minor, but remember Walsh's rule. Walsh's rule is the only time an exception occurs is in your thesis, right? So these are things that can occasionally happen. A much more interesting case is we have this comparison of Ka to Ks. Ks being putatively neutral and therefore a good arbiter of what the basic mutation rate is like before it's filtered by selection. You can have cases, though, where there's a lot more selection there than you think at neutral sites. And a fun paper was this paper in Drosophila, where they basically showed that there's a lot more constraints on third base position than you might imagine, and those constraints aren't related to codon bias. They speculate they're related to maybe stability or splicing or microRNA binding. And a really interesting example of this, you look at the BRCA, the breast cancer 1 gene. So you look at the BRCA gene and you compare humans and dogs. What you see is a window around position 200 to 300 shows a relatively normal level of Ka, but in that same window, across the whole genome, Ka is roughly the same. In this window around position 200 to 300, while Ka is relatively constant, Ka plummets dramatically. So in that window, you get an elevated Ka to Ks ratio, not because of apparently adaptive substitutions, but rather because much more stringent constraints on the third base positions in that window. Probably something to do with binding or regulation. So we think of Ks as being kind of a neutral standard, roughly the same across a gene, and so when the Ka to Ks ratio gets elevated, it's clearly because Ka gets elevated. But it could also be that Ka is relatively flat and Ks takes a nosedive in one region, in which case you get an elevated Ka to Ks ratio. So Ka to Ks is usually viewed bigger than 1 as a gold standard for evidence of selection. I think I say here, a little more humility in its use may be in order, right? So again, it's pretty good, but just be aware that there are exceptions, and it's usually pretty good, but be aware these other issues can arise. Just kind of says what I said before. So I told you about the experiment of looking at MHC. If you look over the entire MHC, Ka to Ks is much less than 1. If I have specific residues I look at that I know are important, Ka to Ks is bigger than 1. How do I extend that to scenarios where I don't know the protein folding structure, or I don't know the subset of functional sites to constrain? The way you do that is you extend it to a codon by codon base analysis. When you have a phylogenetic tree, there are 2 schools of thought which you can look on that tree. Number one is parsimony. How many people have heard of Occam's razor? When in doubt, go simple, right? My thoughts on Occam's razor is you want to use it to shave, not cut your throat, right? And so parsimony basically says, I look at the minimal number of changes you can have to make it work. Sounds pretty good, sounds pretty reasonable. Likelihood is this more formal approach we talked about building a model and estimating parameters on that model. So here's Joe Felsenstein. Hey, look, he's at Nimbus. Look at that. And one of Joe's key early observations is under parsimony, long branches with lots of divergences, tend to attract each other. And so because of that, Eupheus felsensteini has a moth in southern Arizona with a long branch on the forewing. So I named it after Joe. It's Eupheus felsensteini. One of the things likelihood models can do is they can basically account for the fact that we have a bias where transitions and transversions may occur at different rates. So you can build that into the model. It's not built into parsimony. Parsimony also, you're basically constructing ancestral states by minimal number of changes. In reality, there's a probabilistic model under there, and instead of a single value, there's a distribution. Likelihood captures that. So we basically talked about putting these models together. Here's the basic model. We talked about this earlier. I have a specific codon. I've got transitions with a T, transversions with a V. The thing with the star here, everything with the star is a change in the amino acid. So I then multiply that rate by some amount, omega. And the omega basically is what's the rate of replacement to silent. I can then go in for every codon in a protein. So I've got a protein. Position 4, I can then do a likelihood model for codon change over that phylogeny at position 4. And I basically have this, which allows for replacement, silent, and transversions. Then I can basically ask, when I fit my model, is that estimate of omega for that codon bigger than 1 or less than 1? These are the first categories of models. Goldman and Muse, Spencer Muse, and then Goldman and Yang were the people that put these together. And those models then, you have this giant 61 by 61 transition matrix Q, where I label each of these codons by a number. And that's, for example, suppose that's codon 4, that's codon 6. There's the value in this matrix. We can then ask, what does that pattern look like after time t? So what's the probability the codon is I at time t, given it's j at time 0? I can write this as this expression here, where Q is that transition matrix. I can then write this expression here in terms of the eigenvalues of that. I can basically then solve for these certain things. That's what likelihood does under the hood. So that model of having a constant value of omega at a site, and you assume when you fit over the entire phylogeny, it's the same, that's a poor model. An improvement is to imagine you've got 3 possibilities at a site. This is what's called a mixture model. With probability 0, it's neutral, so omega is 1. With probability P of D, it's deleterious. In this case, it's set to a value of 0. And with probability PB beneficial, it's then set to some other value estimate that's bigger than 1. So it replaces that single value for a codon, for omega, by 3 possible values. Totally deleterious 0, neutral, or advantageous with the same amount. You can then fit this model, again with likelihood. People improved on that, and the way they improved on it is, again, you've got 3 classes, but now, if it's deleterious, you draw from some distribution where that value ranges for a given substitution from 0 to 1. If it's advantageous, you draw from some distribution, usually like a truncated gamma, where value ranges from 1 to infinity, and basically then you can basically assume what these are, and then estimate, for example, the mean here and the mean there, and fit all those by likelihood. So this is what likelihood does. It makes more complicated models. So you can basically fit those, and then go back and then estimate these various components. And we'll talk about that in next lecture about what those estimations are. So you can take these codon data and you can ask what fraction of sites seem to be advantageous, what fraction are beneficial. If they're advantageous, how strong is omega? And we'll see next lecture how to relate omega to the average strength of selection. The other question you can ask is, when I run a model and I get a significant result that selection is important, the next question is, which are the critical residues that are important? Well, you can then use Bayes' theorem for them. So what Bayes' theorem does is it flips the conditional. I can basically ask, if I have data in a certain class, for example, deleterious, what's the probability, given my model, of getting the observed phylogeny? I want to flip that around and say, given the observed phylogeny, what's the probability I'm deleterious? So Bayes' theorem is a classic way of flipping conditionals. Let me give you a good example of Bayes' theorem at work. So in southern Arizona, we have a lot of Native American reservations, and almost all of them have casinos. It's only fair we stole their land, now they're stealing our money back. Perfectly, I love symmetry. So suppose you go into one of these casinos, and you walk in the door, and there are two people playing the slot machine. One of them just hits, gets all excited, gets a huge bucket of quarters and leaves. The other person throws down their hat in disgust and says, I've been playing this machine for 6 hours and I haven't gotten a single thing. And your choice is, you've got two machines that just became open. Which one should you play? Should you play, don't get excited here. Should you play the machine that just won? Don't answer yet. Should you play the machine that hasn't won because it's due? Or does it matter? So how many says play the machine that wins? How many says play the machine that loses? Don't look around, raise your hand, be honest here. By the way, everyone's wrong so far. How many say it doesn't matter? And the answer is play the machine that wins. Oh, what? That's Bayesian statistics. Here's why. You're making an assumption you don't realize. Most of you are correctly assuming on this machine here that loses, each poll, the win probability is still the same. Say it's 1 in 50. That person had a run of bad luck. But their run of bad luck doesn't change the fact the next time you pull that handle, you have a 1 in 50 chance of winning. So the fact that it hasn't won doesn't change its intrinsic chance of winning. The machine over here, just because it's won, that doesn't change the chance of winning on the next pull. So right away you think, oh, well, it doesn't matter. The answer is it does matter because the intrinsic chance of winning could change. You have a 1 in 500 chance of winning each poll, independent of what happened in the past. You have a 1 in 20 chance of winning each poll, independent of what happened in the past. If all you observe is one machine wins and one doesn't, which one should you play? The one that wins. That's the best information about which has a higher intrinsic chance of winning. If you want to be a degenerate gambler, the machines actually post the odds on them. And also, if you go through Vegas, it's almost always the case, you have these, if you want to get from your hotel outside, they have you wind through the casino. Instead of walking 10 feet the door, you have to wind through this elaborate, and you walk by all these machines. They always put higher winning machines on the edge. So as you're walking by, you hear winning and figure that could be me. So that's an example of Bayes' theorem, is you're using prior information. That's the kind of deep example. There's a great book called The Theory That Wouldn't Die. It's a very easy read that tells about Bayes' theorem and using things like breaking the Enigma code and finding missing atomic bombs and hunting submarines and all sorts of really interesting things. But the basic idea behind Bayes' theorem is you flipping the conditional. One conditional may be easy, other conditional may be hard. And so if I tell you what category you're in, I can use the model to generate the data. What I want to know, though, is given the data, what's the chance my codon is in that category? Bayes' theorem basically tells you how to do that. So, for example, if I want to ask what's the posterior probability that a given site is advantageous given the data for that site, I then take what the data for that site would be if omega is bigger than 1 times the chance I estimate after the fact of being an advantageous site, and this is being deleterious, neutral, and advantageous, and that's called a Bayesian posterior. We can basically use that then to put posterior probabilities on specific sites. That site has a 90% chance of being an advantageous, having advantageous substitution. That site has a 30% chance of having advantageous substitutions. I mentioned the example earlier about Erebus and looking for functional sites in it. So that's kind of your quick overview of these methods. We're now, after the break, going to take these methods now and use them to ask what fraction of sites are advantageous or deleterious in genomes. And so we're going to be using this same machinery we have here. So covered a lot of things.